Okay, our second category would be the carbohydrates. Carbohydrates are diverse. They play an important role in all living things. Primarily, we speak of carbohydrates in terms of their energy. Generally, they are the first source of energy for all living things. They are also an important component of nucleic acids. So we'll look at the sugars that make up or help to make up DNA and RNA. And they help to form the bacterial cell wall. So when we study bacteria in the next couple of chapters, we'll talk a lot about peptidoglycan. In speaking in terms of carbohydrates, it's the glycan portion of peptidoglycan that is made up of carbohydrates. So you'll hear that again very soon. Carbohydrates contain a carbon, hydrogen, and oxygen ratio of 1 to 2 to 1. This is a general rule of thumb. Sometimes it's not exact, but CH2O is the general formula for any carbohydrate. So for example, glucose is C6H12O6. There's a 1 to 2 to 1 ratio of carbon, hydrogen, and oxygen. So you could easily identify that as being a carbohydrate. Monosaccharides are the monomers of carbohydrates. So make sure that you know that monosaccharides are the generic terms, or is the generic term we use referring to the carbohydrate monomer. So proteins, the monomer were amino acids. For carbohydrates, the monomer, monosaccharide. And saccharide means sugar. So all sugars are carbohydrates. Monosaccharides can be classified based on their size. They are either 5 or 6 carbons. And the most common ones are going to be your 5-carbon sugars, ribose and deoxyribose. So again, we'll see those in nucleic acids. And then your 6-carbon sugars like glucose, probably the one we talk about the most. Glucose being a 6-carbon monosaccharide. Okay, and just some pictures of some 5-carbon monosaccharides, ribose and deoxyribose. You could count the carbons, just different ways of representing the structure of the molecule. Disaccharides, di meaning 2. So a disaccharide is nothing more than 2 monosaccharides covalently bonded through dehydration synthesis. Again, something we looked at in the protein video. Some common examples, lactose and sucrose. We'll skip that. Polysaccharides, poly meaning many. Polysaccharides, at least the ones listed here, are made up of glucose. So all of these are made of glucose. And again, the word subunit implies monomer. So cellulose, glycogen, and dextran are all large sugars or polysaccharides made of glucose subunits. The difference between cellulose and glycogen is really only in the arrangement of glucose, but they are just large chains of glucose. Cellulose is the most abundant organic molecule, or one of the most abundant organic molecules on the planet. Cellulose makes up the cell wall of plants, but for our interests in this course, cellulose is the primary component of the cell wall in algae. So we won't be studying plants so much in here as we will be studying algae later in the year. Glycogen is how animals store glucose. Some bacteria use glycogen, but if we're talking about us as humans, we are storing our glucose in the form of glycogen. So cellulose is primarily a structural polysaccharide. Glycogen is a storage polysaccharide. One that you may be unfamiliar with is dextran, and this is a storage polysaccharide in bacteria. This is how bacteria would store glucose. Looking at nucleic acids, so moving to our third category, we're only going to look at DNA and RNA, and this is something that we reviewed very early in the year, so this should not be brand new. DNA and RNA, the generic term for the monomer would be nucleotides. So nucleotides are the monomer of nucleic acids. So proteins, amino acids, carbohydrates, monosaccharides, nucleic acids, nucleotides. Looking specifically at DNA, all living things, all living cells contain DNA. Every single cell. You will need to know the parts of the DNA monomer. So all nucleotides in DNA contain a phosphate group, contain the deoxyribose sugar. So there is our carbohydrate component, the 5-carbon deoxyribose, and then one of 4 nitrogen-containing bases: phosphate, deoxyribose, and base. Your base pairing rules: adenine and thymine are always paired together. Guanine and cytosine are always paired together. So we use the term complementary, meaning that they pair together, like pieces in a puzzle. Adenine and thymine are joined by hydrogen bonds. Guanine and cytosine are joined by hydrogen bonds. Recall that DNA is a double-stranded molecule, so the 2 strands are held together by these hydrogen bonds. The sugar-phosphate backbone of a DNA molecule, phosphate sugar, that makes up the outside part of the molecule, the internal component of the molecule, the bases, again held together by weak hydrogen bonds. Looking at DNA versus RNA, we looked at this again briefly earlier in the year when we reviewed transcription and translation. RNA is different in that it contains uracil instead of thymine. RNA contains ribose as its sugar, and it is a single-stranded molecule. Those are the big differences between DNA and RNA. RNA is used in the process of protein synthesis. And so when a cell needs to build a protein, it has to use its DNA to make RNA, RNA being the recipe for the specific protein, and then we would use the RNA to assemble the amino acid sequence of the protein. So again, the relationship between building a protein and DNA and RNA. You need DNA and RNA to ultimately build any kind of protein in the cell. And our last category would be lipids. Lipids are a critical component of the cell membrane. And so when we visit or revisit the cell membrane shortly in the bacterial chapters, we'll talk about the bacterial cell wall having a peptidoglycan component, but all cells have a cell membrane, and it's really the same regardless of the cell type. Lipids would be the smallest category of macromolecules or organic polymers. And one of their defining characteristics is the fact that they are non-polar or insoluble in water. So lipids do not dissolve in water. So we'll look at simple versus compound lipids. Simple lipids in particular, contain only carbon, hydrogen, and oxygen. They are different from carbohydrates in that they have lots of carbon and hydrogen and only a little bit of oxygen. So there is no ratio like we found with carbohydrates. I would like to focus on saturated versus unsaturated fats when we're speaking in terms of simple lipids. So if you look at this picture, a saturated fat is one that contains all single bonds between carbons. So you can look at this chain and you would only see single bonds between carbons. An unsaturated fat has at least one double bond. Okay, so that is really what defines saturated versus unsaturated. Saturated, all single bonds between carbon. Unsaturated, at least one double bond. Saturated fats are the ones that are solid at room temperature. They come from animals, whereas unsaturated fats are liquid at room temperature and come primarily from plants. So your oils are generally unsaturated fats, like olive oil, peanut oil. Steroids are also considered simple lipids. They are unique in that they have a ring structure. We classify them as lipids because they are insoluble in water or nonpolar. And so the one example of steroids that you should know would be cholesterol. Okay, the word sterol is in there. Compound lipids, we're just adding to simple lipids. So we have our fats. We've got a saturated fat and an unsaturated fat making up a phospholipid. So phospholipids are extremely important because they make up the cell membrane of all cells. The lipid component of the phospholipid would be the saturated and then the unsaturated fatty acid tail. And then, of course, the phospho part would be the glycerol molecule containing the phosphate group. So sometimes we just say the phosphate head, that's fine, and then the fatty acid tail. And that's really all I'm going to ask that you know. I'm not going to really get into your knowing the glycerol, as long as you know that the head of the phospholipid is the phosphate group, and then you've got the two tails, one being saturated and the other being unsaturated, because see, double bonds. Phospholipids, again, make up the cell membrane. The cell membrane acts as a barrier. It controls everything that comes in and out of the cell. So extremely important in most cellular activities. And even within the cell, if we're talking about eukaryotic cells with membrane-bound organelles, the mitochondria, the chloroplast also feature phospholipid membranes. Okay, so please make sure that you've read Chapter 2. It should be mostly a review of what you've done in biology. I do know that it's been a while for many of you. If you have questions, please come see me, because this is all we're going to do in terms of talking about the 4 organic families.